So I wanted to start by talking about my background and how I ended up as a professor at Johns Hopkins. And so it wasn't really that long ago that I was like you guys, where I'd go to an assembly in middle school and high school, and I'd hear different people speak. And I was interested in science since a young age. I was always very curious with the world. And so with the early years, I was very curious about the world and science. And so I was born in Canada, and I grew up in Miami, Florida. So I'm a first-generation American. And I was very interested in science, and then there were these magnet schools, public schools you could apply for if you're interested in science. And so there's one in Miami called Mast Academy, which was for marine and atmospheric science and technology. So I thought, oh, that'd be a perfect fit, and I applied after middle school, and I got in, and I was so excited because I could take classes like marine biology or solar energy in high school, and I really liked that. And I got involved in other extracurricular activities, which I'm sure many of you do too. And so in high school, one of them was Mu Alpha Theta, and that was really great. And then another one, though, was US First. And that one was about learning about engineering and then creating things. And that was something that I didn't really know a lot before. And I thought I wanted to be a scientist, marine biologist. But then when I learned about engineering and how engineers build things and create things, that really intrigued me even more. And so when I went to college, then I wanted to learn more about engineering. And so I went to Carnegie Mellon in Pittsburgh for college, and I also spent a year at Imperial College in London, and I really recommend study abroad if that's something that you guys think about as you go to college. But in choosing a major, it was hard to decide, what I wanted to do. I knew I wanted to do engineering, and I liked biology and chemistry. And actually, when I went to college, BME, Biomedical Engineering, wasn't a major actually you could take on its own at Carnegie Mellon. I later, for my PhD, went at MIT. It wasn't a major you could take there for undergrad. It was still being developed as a major. So some schools like Johns Hopkins had it, but a lot of schools didn't. Nowadays, a lot of schools have biomedical engineering as a standalone major. But what I'd encourage you to do is when you go to college, if you're really interested in an emerging area, you can major in whatever is close to that and then take extra classes or a minor or maybe a double major to then explore that area. And then in the future, that'll become a big discipline. But if it's in the early days, you can still take the classes or try to do undergraduate research to learn about that area. And so chemical engineering, if you don't know as much about chemical engineering, you design products and processes, and a lot of it could be processes that make toothpaste or that make semiconductor chips. And so I was initially in chemical engineering, and I went to the different kinds of career fairs to learn from the alums what they do. And so there was someone that was there that worked for a big consumer products company, and I was asking him, well, what are you most excited about that you're doing out there in the real world? You got this engineering degree from Carnegie Mellon, and what are you doing? And so they were saying how he was very excited about this product he was doing with toothpaste. And what it is after you guys use a toothpaste and you brush your teeth, how do you know your mouth feels clean, or what do you feel like it's a good toothpaste? Anyone? Maybe you slide your tongue over your teeth, and if it glides, you think, oh, my teeth feel pretty clean. And so that's how it works in a company. They go to people and ask them for feedback, and then that's the feedback they get. The focus group says, oh, when I brush my teeth, if my tongue glides over my teeth and it's very smooth, I feel refreshed and like it's clean. And so that was then the project is, well, how do you make that happen? And so with engineering, if that's the goal and it's for the company, what it ends up being is actually depositing a surfactant, kind of like a soap. It doesn't taste bad like soap, but the same idea on the teeth from the toothpaste then makes it that when your tongue glides over your teeth, it goes really smoothly instead of getting stuck. And so that's what's in the toothpaste, and then the people try out the toothpaste, oh, this is great toothpaste, and the sales go up. And so that works, and that makes money, and everyone makes, but I was like, I didn't go to school for all these years and all the late night homework assignments to do that, to deposit things on people's teeth. I don't want to do that. And so I was thinking, well, how can I work on something that the things I'll make are going to definitely help people? And so biomedical engineering was really what that was. And I started to do undergraduate research in gene therapy when I was in college, and I really liked that. So then when it came time to go to grad school, I realized I wanted to continue to do research. After that undergraduate experience that went really well, I wanted to keep doing research and then wanted to go to grad school. And so I went to MIT for grad school to major in biological engineering. And then I also did a postdoc in chemical engineering after that. And what I learned is so many different things of how to approach a problem and different kinds of techniques and nanoscale and using drug delivery devices and biomaterials and health. And it was really great. I was in a really big lab The Langer lab, I don't know if well, you can see on the screen that big photo, that's one lab. That's the largest biomedical engineering lab in the world, pretty much. It's more than 100 investigators. It's Bob Langer's lab, and it was a wonderful environment to learn and then develop myself as a scientist. And so that started to steer me towards being a professor myself after having that background and really liking working with other students, learning and researching new things. But something I wanted to say along the way, and this is important in high school, in college, and grad school, is your mentors. And so different people that give you advice and tell you about where your path can lead and about your specific stuff you're doing on research, but other things in professional development. And so my advisor for my PhD and my postdoc is Bob Langer. And this guy is an amazing man. If you haven't heard him speak, you could look on YouTube and Google it. He's the most cited engineer in history. He has more patents than anyone else other than Thomas Edison, but he's still alive and inventing, so he's probably going to pass that too. And then he's won different kinds of awards, like he just won a £1 million award from the Queen of Elizabeth. He's gotten medals of science and technology from both Democrat and Republican presidents. But he's also just the guy that invites you over to his house or to the beach just to hang out. And so, I learned a lot about him, both in terms of the science side and leadership side, but also, how to live life. And so from graduating there, I then went to become a professor. So to become a professor, you need a PhD if it's at especially like a research university. And then I had a short postdoc in between. A lot of times in biomedical science, after your PhD, you do some more research, and that's your postdoc. And so my primary appointment's in biomedical engineering. I'm an associate professor of biomedical engineering, but a lot of the work we do is at the interface with other departments, and we really like to collaborate with other scientists. And so that's what we do. And I now have appointments in other areas like chemical and biomolecular engineering, material science and engineering. And then on the medicine side, I have appointments in ophthalmology, so that has to do with eyes, neurosurgery, the brain, and then oncology, cancer. And those are the different applications. We want to invent new tools and technologies to help people live better, healthier lives, and particularly to look for cures for blindness, for diseases of the brain, and for cancer. And so, where did I go then with my lab, and what was I interested in? So I was always interested in comic books and video games and movies and things like that. And so a lot of our research actually is at the interface with this. So if you think of some different movies, like with the X-Men, they have all these different powers. How did the X-Men get their powers? And mutations, right? So they're born with these mutations and sort of feel different from other people. They have this mutation, it gives them these powers. There's other superheroes, like Spider-Man and Incredible Hulk. They weren't born with that though, right? Something happened to them in their environment, like bitten by a radioactive spider or a genetic spider, or had a radioactive blast, and then they get all these powers. So again, it's these mutations, but now it's not inborn. It's something that you develop over time from the environment, things like radiation. And then in other movies, so Captain America, or you may know this other guy, Solid Snake from the Metal Gear video games, then these are sort of these super soldiers. And how did that happen is they had this was now by choice, not this sort of random event, that they had the serum that then changed their genes, and in this case, in the movies, we give them strength or heightened senses or extra kind of powers like that. And so that's what's in sort of Hollywood and then this idea. But the ideas of mutations is not just something that's in Hollywood. Having the mutation let you shoot laser out of your eyes is in Hollywood, but there's mutations, all around us. And so as you may know, what this has to do with is just a gene that then has a change, and then that changes its function. So the gene stores the information, and then it encodes for protein, and then that's what does the function. And so if the gene has a mutation, the protein has a mutation. And so this is something that could be from birth. It's something that could develop over time from the environment. Like you get a mutation, it could lead to cancer. And it's something where we can see mutants. So for example, this cat, if you look at its paw, it actually has 6 toes instead of 5. So it doesn't necessarily run faster or climb better because of that. But if it did, that's something that over different generations of natural selection, you could have, all cats could end up with then an extra toe. If there's something like that, that's how evolution can happen. And so there are these mutants that happen. More normally, though, what ends up happening is with the mutation, you lose function of something. So your protein now might have a disease. It doesn't shuttle cholesterol as well, and you might get heart disease. It might be a mutation that makes your cells grow uncontrollably, and you get cancer. So a lot of times, the mutations end up causing diseases. And so that the diseases that we have that we treat with a pill or something every day actually could have a genetic component, and we're just treating the symptoms of the disease. The disease still has this genetic component that we're not able to treat. But in the near future, it's something that we could treat. And this is the idea of gene therapy. So it's using genes as a therapy in genetic medicine. And so the gene itself could be as a drug. And so that gene, that DNA, codes for mRNA, and then through transcription, mRNA is made, and then translation, the protein is made. And so in the near future, we could think about genetic medicine that could go to the root cause of that disease and treat it and potentially even cure it. And this could be things like cystic fibrosis or hemophilia, or things like even cardiovascular disease or cancer. But there's a famous saying in the field, there's only 3 problems with gene therapy. It's delivery, delivery, delivery. It's like with real estate, there's only 3 important things: location, location, location. The hard thing is, how do you get that gene safely and effectively to the right cell that it can work and not damage anything else? And so the 2 approaches have been viral and non-viral. Viral is using a virus. And the challenge is there. So viruses have evolved to be good at infecting and passing on their genome. Actually, if you look at any of your DNA, you have all sorts of segments of DNA that come from viruses that have evolutionarily been in our genome. And so viruses continue to do that. And so the idea is you take some of the viral genome out, put in the good gene, infect, and then you can deliver the therapy. But the problems is sometimes it could cause cancer due to a mutation that it makes itself. It could lead to a severe immune response that could be even deadly. And so there's a lot of work going on making this safer, but our interest was, well, how can we make it safer by not using a virus? And so we look at viruses, and you can see some of the different viruses on the slides there under biological. You see those different shapes. And then the biomimetic are ways that we mimic nature by making particles, nanoparticles, that then have the same kind of size, same kind of shape. They can have very similar properties to the virus, but they're well controlled. They're not going to cause a strong immune response. They're not going to cause cancer. They're going to degrade in a safe way, and they're not infectious. And that we're going to make these as sort of these synthetic virus-like particles, but they're not viruses at all. They're just mimicking how they work. And so that's something in engineering we try to do, is to learn from nature and then design new systems that mimic aspects of that. And so what we do in our lab is we design new types of polymers. And so polymers are basically plastics, except this isn't like a plastic that you think about if it's thrown out in the environment, it's there for years and years and never degrades. These are new kinds of plastics that degrade in just hours when they're in water. And they break down as safe little molecules that can be metabolized by the body just like sugar is. And so nothing's left behind, and it's very safe. It's like if any of you have had sutures, that then the doctor leaves in and they biodegrade, they don't have to be taken out again. It's just like that. It just gets absorbed by the body and broken down. And so we're going to use that kind of technology, but to make really small nanoparticles that can then deliver these genetic medicines. This is what the polymer looks like. It's not important too much about the structure, but just the idea that we design these polymers to have positive charge, so they bind to negatively charged DNA, and that positive negative, they interact together and they self-assemble into the nanoparticle. But the problem is that the current polymers, like this 25 kPa on this graph, are very low at delivery compared to the virus, which works much better. So how do we make that work better? How can we design the polymers to safely and effectively deliver these genes? How can we engineer this technology? And so what we do is we look at the different steps in gene delivery, that initially the polymer and the DNA have this electrostatic interaction, the positive and the negative charge. They come together to form this nanoparticle. The nanoparticle needs to be taken up into the cell, and this is endocytosis. It's like the cell's eating it. And just like when we eat our food, we digest it in our stomach. When the cell ingests this particle, they degrade it in this endosome and lysosome. And so we needed to design the particles to get out of that degrading space into the cytoplasm, which is the real inside of the cell. And so we designed the polymers to do that. And once in their cytoplasm, we then designed them to release that nucleic acid and then break down safely and not cause any toxicity. And then the DNA makes its way in the nucleus to be expressed. So those are the different steps. There's a movie here. I don't have a mouse right now, so I don't know if someone can maybe hit play on
My
the
name
movie.
is Jordan Green, and my lab at Johns Hopkins is really interested in how we can develop new types of technologies to treat devastating brain cancers that even with the best surgery, chemotherapy, and radiation still has poor patient outcomes. We've now developed a new technology that has a promise for treating this disease in a new way. The challenge with brain cancer and cancer broadly is that cancer cells were once upon a time normal cells that have now mutated and grow uncontrollably. Here, let's see what we have in the toy box here, some dominoes. We can illustrate this concept. So if we have some normal cells, these are these black dominoes here, and cells are arranged next to each other in the body. They're normal and they're happy, but what could happen is there could be mutations that part of it could be genetic, part of it could be environmental, where some of them change into these cancer cells with different colors that aren't normal. And now you have these cancer cells and you also have healthy cells. And the way conventional chemotherapy works is it can't really tell a difference between a healthy cell and a cancer cell. It's really a poison, a toxin that'll kill everything. So if the cancer chemotherapy comes through, it'll kill your healthy cells and get your cancer cells. And this is why patients might lose their hair and have nausea and all these other complications. And so what we'd like to do is to attack just those cancer cells and not hurt healthy cells. Let's imagine that these balloons are cells. So we've developed a technology to send instructions to these cells to get them to do whatever we want. This is a little bit like how an electrical engineer could program a computer. As a biomedical engineer, we want to program cells. And we especially want to program cancer cells differently than healthy cells. This is our mission impossible. We were able to use this new technology to deliver our instructions to cells to make them change shape or color. And using the same type of technology, we can actually have the brain cancer cells self-destruct altogether. This approach is called gene therapy. The instructions that got the cancer cells to do what we wanted them to do, those instructions come from a nanoparticle, which is a particle made up of plastic and DNA. And it's about 1,000,000 times smaller than this particle here. And the DNA is inside of it. And what it does is it can safely deliver this cargo through the different barriers and then let that information go to the right cell. So the cell does what we want. We've demonstrated this now in human brain cancer cells in a dish and in live mice. And we're really excited about the next steps as we move this forward to patients. This type of discovery is possible here at Hopkins due to the natural collaborative atmosphere, where myself as a biomedical engineer, I can work closely with excellent neurosurgeons, and together as a team we can innovate these new technologies that can make an impact to patients.
All right, so I hope I showed there with some of those toys, the aids, that what we want to do is then engineer the cells to be able to find target cells, and then we can program them just like a computer engineer programs a video game. We program those cells to behave in a new way, and particularly to heal them. And so if it's a cancer cell, we'd want it to self-destruct. If it's a healthy cell that's diseased, we'd want to heal it so that that protein now is repaired. You don't need to take a pill every day the rest of your life because it's now fixed. And so that's the idea with genetic medicine. And so when we say nanoparticle, it's a particle that's really, really small. And how small is it? Well, a globe of the Earth to the whole Earth, the globe is about 10 million times smaller. And then if you look at that globe, about 10 million times smaller than that is the nanoparticle. So it's really small. And then, so one of the things we look at as engineers is the different sizes and shapes of different things in the natural world, from millimeter sizes down to atomic sizes. And then how can we create new kinds of materials that then mimic, again, sort of the natural biological materials. And there's different technologies that are going on right now with engineers designing new types of particles with different shapes, from the top down by molding them in different ways, or from the bottom up by having this self-assembly. So the nanoparticles that we're talking about with these positively charged polymers, negatively charged DNA, is this bottom-up self-assembly, where the molecules interact together and then build the particle. And so nanotechnology and nanobiotechnology can deliver a lot. It can deliver new types of sensors, new types of medicines, and new types of gene therapies among the many applications. these are the specific polymers that we've made in my lab at Johns Hopkins, and we've designed them to be biodegradable. The chemical formulas don't matter here, but the point is that they're responsive and they break down. And so that's what the arrows show. The first set of arrows shows the synthesis, and the second one shows how they break down in the body, which is important so that they're not toxic and so that they release their cargo. But how do we measure if this gene therapy works? Well, so the DNA, the sequence of it, doesn't matter for the delivery technology. Once you create the right delivery technology that's going to work, one of the really great things is you can switch that drug, the gene, whatever the sequence of the bases is, doesn't matter. The technology will work the same, be just as effective, and you can change that sequence. So what we wanted to do then was to deliver a sequence that then we can measure, oh, is it working or not? And so what we did is we looked, other scientists have studied the jellyfish and see that the jellyfish has certain protein that then fluoresces green. So if there's blue light that's there, it'll shine this green light back. And so what we want to do is deliver the DNA that coded for that protein, this green fluorescent protein, and then if the DNA delivery worked, we would see them glow green. And then we knew that the process works and the technology has been validated. So we wanted to do that. And then how do you measure it? You can use a microscope or these different kinds of plate readers or something called a flow cytometer to measure the fluorescence that could come from these cells. And we find that it does work. So if we looked at different cells in the eye or in brain cancer and we deliver our DNA particles, if the DNA goes inside, goes into the cytoplasm, goes in the nucleus, gets transcribed, translated to protein, it'll glow green. And that's what we see here. It's fluorescing green. And this shows that it works in a dish. What about in an animal? You can't see this fluorescence that well through an animal, but what you can is with a firefly, there's a different protein, it's called luciferase, where if you deliver that one, you can then see this luminescence and light come from the animal. So if we inject the particles into a mouse, and then we change their coding, we can have them go to different organs. And so what you see on the bottom here is different organs lighting up, like lung or spleen or liver, because we're getting the DNA to different tissues specifically, and then it's emitting the light. Now, instead of having it light up, though, it could be delivering a protein to cure disease. It could be, if it's a cancer cell, delivering a protein to kill that cancer cell in a way that only targets that cancer cell and not a healthy cell. And so that's what we wanted to do. And so in a dish, we have that here with healthy liver cells, and then liver cancer cells are the red ones, and they grow together in the co-culture. And we wanted to deliver the DNA and see could it just go to the cancer cells, the ones that are always red, and not the other cells you see there that aren't red. And if it works, they'll turn green. And that's what we see is we can get it to go to just the cancer cells and turn them green, and not the healthy cells, which are sort of in the background there that aren't being transfected. So that shows, again, this proof of principle that this can work and it can be specific. And then where would it be interesting to do this would be in the brain. So there's this awful disease, glioblastoma, which is stage 4 brain cancer, where even with the best surgery, radiation, chemotherapy, patients only live a little longer than a year. And we wanted to see, can we get those brain cancer cells and transfect them? And so we've done this with different brain cancer cells, including brain cancer cells that come from patients that donate those cells for science to see if we can develop new kinds of therapies. And we can transfect them really well in a dish. And then we wanted to see, well, what about if we look at brain cancer cells in an animal? And so this is actually rat brain cancer in a rat, and we inject the particles and they spread through the brain tumor, and you can see it light up with green. That's showing that it's delivering the gene in that animal that has brain cancer to the brain cancer, and it's going to just the brain cancer. But what we really want to do is to kill those brain cancer cells, not just light them up. So then we change the DNA to be instead a protein that if it's expressed in the brain cancer cells would lead to those brain cancer cells dying. And so we did this in the rat model. This is a survival curve, which shows how long the animals live. And this is a really aggressive cancer for the rats. So you see all these curves sort of ending at day 15. They only last about 2 weeks in this model. The brain cancer is so severe. But then with the treatment in the red curve, you see that it lasts an extra week, which is 50% longer. So this isn't a cure, but it's showing that in an animal model, we can inject these particles, they're safe, they can deliver the gene can cause those brain cancer cells to die, and the animal can live longer. And so we're working with clinicians at Hopkins to design better therapies using this approach to then go after cancer in new ways. And the great thing with this is, unlike chemotherapy, which is a poison that goes everywhere and kills healthy cells too, we can be specific to just those cancer cells. And so I hope I've shown how biomedical engineering can enable new technologies for human health, and that these technologies can be used to reprogram cells to function in new ways, including to kill cancer cells or to heal disease cells. And that what's important is that this collaboration with others, in this case in medicine, and then along the way to how I got this position and with my students, mentoring and meeting people and networking is really important too. So it's not just the science, but also sort of the people skills and working with people, because this is all done in teams. And then science leads to unexpected things, unexpected results. And then another thing, I was invited to then get to, these are all scientists and engineers that were all invited to go to the White House last spring to meet with President Obama. And he talked so much about how important STEM was, and science, engineering, and math was, and how especially to have a diversity of talent in STEM. And he even gave me a fist bump there, so that's like one of my best moments in my life ever, and science led me there. And so I'd like to thank my lab that did all this work, and then especially also collaborators and funding sources. So NIH and the government actually funded a lot of this work. And then thank you so much for your attention. I'd be happy to answer any questions.